Hello and welcome back. This is the EIT Health Catapult Biotech Semi-Finals 2021. Thank you for joining us. Now this afternoon we have a very big program and we will be leading, of course, to the big moment at the end of the day at 5 o'clock with the awards ceremony. That will be preceded by our third expert insight. But before that, we have two more pitching sessions. The first session takes 4 pitches, and the fourth session, which will take 3 pitches. And it's the same format as this morning: 5 minutes pitch, 5 minutes Q&A with our panel of jury members. And we are strict with the timing, so if there is an overrun, I will have to do that horrible job of stepping in and cutting you off. So, before we begin, a word, a message to all of the pitchers this afternoon. Have a fantastic time, enjoy your session, have a great pitch. So let's begin. Let's go straight now to Switzerland. And we are going to meet Luc Henry from the startup Limula. Luc, it's over to you. Luc, I think you might be on mute. Can you unmute, Luc? We can't hear you for the moment. And we would love to hear you. So, we still cannot hear you. All right. Can we try to reload the page or to refresh the page?
Hope you hear me. Can you?
We can hear you now, Luc, loud and clearly. Great. It must be those mountains in between us. Okay, fantastic.
So you can hear me now? Yes? Okay. Thank you very much. So good afternoon, everybody. My name is Luc Henry, and I'm the co-founder of Limula, based in Lausanne, Switzerland. And I believe cancer patients should all have access to the very best treatment available. But before I tell you about Limula, I'd like you to meet Emily Whitehead. Emily was diagnosed with leukemia when she was 5 years old. Less than a year later, she was close to dying and fortunate enough to be the first child to receive an experimental drug called Kymriah. Emily is now 16 and her cancer has been gone for more than 9 years. Kymriah isn't just any molecule. It's a living drug made of genetically engineering the patient's own immune cells. I can hardly think of a treatment more personalized than that. CAR T cells reached the clinic only a few years ago, and I would like to insist on the fact that they are nothing like what was available before. Cancer patients who would survive only for a few weeks can now be cured with a single dose. Today, there are 5 CAR T cells therapies available and 2,000 in clinical trials. The FDA estimates that 100 could be on market in 2030. This treatment will be used against a broad range of cancers with the potential to cure over 2 million people each year, leading to $82 billion in global annual revenues. Unfortunately, we know that most of these patients will not benefit. The science works, but the economics doesn't. The problem is this: $475,000 is the price to pay to receive these life-saving drugs. Cell therapies are so difficult to make that they are totally unaffordable. Less than 20% of the patients who could benefit will ever receive a dose if nothing is done to develop a new manufacturing approach. At Limula, we address this gap with a solution that enables CAR T production at scale. Manufacturing is now centralized. We'll bring it to the hospital. The process is manual and time-consuming. We'll make it automated. It is unreliable and expensive, will make it robust and cheaper. CAR T-cell manufacturing currently requires several instruments and 2 weeks of hands-on procedures. It's an open process that is costly and often fails due to errors and contamination. At Limula, we spent the past 2 years working on a solution to bring the complex processing a single device that is fully closed and automated. It is the only way we will transform this incredible scientific and medical success of CAR T cells into a market success for the benefit of patients. This is how the future of cell culture looks like. We have a strong patent covering key design aspects of our solution and all the processes it can support in the field of biotechnology. What is absolutely unique about our platform is that it enables the production of personalized therapies without the cells ever leaving this container throughout the entire process. Working with clinicians from the University of Geneva Hospital, my team already replicated 4 steps central to the production of any CAR T-cell product. The 2 remaining steps, selection and formulation, are specific to a given therapy, and we plan to tailor them to customer use cases when relevant. Our product line will be composed of 2 different versions of the operating unit and a catalog of consumables. X1 is a benchtop operating unit for single batch manufacturing. X-Array can accommodate up to 6 kits in parallel for the routine production of treatments. And because 1 kit is needed to produce a unique batch for each patient, Limula will generate recurring revenues with single-use consumables that can be sold at 10 times the cost of together clinicians and industries in the field, please.
So we've had a slight technical problem there for the sound. Okay, so we have to stop there, but let's open to our jury. Can we go over to the jury? Good afternoon. Would our jury like to open the questions, please? Yes, Renato, please go ahead.
Yes, so being based in Lausanne, I have to confess that I'm positively biased by the old technology coming out from the EPFL. My main question to you is about, it's quite clear the tech development. I would like to see also some industrial application and some market need specific, because you highlighted very well the problem, but I would like to understand if you are connected with the industry in order to solve this big problem, or it's at this stage still, let's say, an university tech project. Thank you.
No, thanks a lot for the question, and apologies for the problem with the connection. You missed my last slide, which was clearly stating that we are in conversations with about 30 companies active in the field of cell and gene therapy. All of them complain about the existing solutions. And so what we're trying to do is to address the limitations of current technologies or of technologies that are currently under development, and try to tailor our solution to specific use cases that these 30 companies and more actually voice to us. So we are deeply connected to the CGT industry.
Thank you. So let's move to a second question. Shilian, go ahead.
Thank you. I'm wondering what differentiates you from companies with a similar approach like Miltenyi or Lonza with the Cocoon? And secondly, what would be your sort of primary focus? Are you also going after adoptive T-cell therapies? Maybe CD19 CARs? Could you maybe comment on that as well?
Absolutely. So again, apologies, it was on my last slide. The thing we focus on at the moment is a proof of concept on a marketed CAR T cell therapy that is an anti-CD19 CAR T cell. So indeed, we want to demonstrate the versatility of our device, but first of all, we focus on a CAR T cell, autologous CAR T cell product as a proof of concept. We are exploring other cell therapies, but we want to first really deliver results on autologous CAR T cells.
Julian, I believe you have
How is
a
this
question.
any different from...
Oh, sorry. Excuse me.
And
Yes.
Are there any differences? Have you noticed any sort of differences in persistence or efficacy between the cells you've manufactured and those of your competitors?
So far we benchmarking against the manual process of manufacturing, and we don't see any difference, or we see even better performance in our device. We haven't yet benchmarked against competitors, but maybe to answer your question about the competition, what we've done is to listen to users of the Miltenyi and Cocoon machines and try to address the limitations of these. One of which I can tell you is the volume of the cell culture. We have an increased volume compared to competition. And also, because we keep the cells in the same vessel across the process, we tend to lose less cell material between steps. And also the cells are handled in a more gentle way. So some manipulation lead to T-cell exhaustion, for example. So T-cells that are suboptimal for reinjection, our device should be able to address this.
Thanks. Julian, I believe you have a question. Go ahead.
Yes, thank you. And thank you for the presentation. So did you discuss any of this with regulatory authorities, and can you share the feedback, if so?
So we haven't discussed extensively yet. We're trying to talk to experts in regulatory rather than regulators directly, because we still have some engineering pain points to solve before we can actually design freeze and think about regulatory considerations. But we certainly want to design a device that is meant for GMP, so that by design it is, and not tweaking our device to GMP requirements later on.
Yes, Lina, please go ahead. We have 15 seconds.
You seem to have a very strong team on the scientific side. Could you comment quite quickly on the business and also on the regulatory and GMP side of your team?
Clearly, the next hires.
Just running out of time. I'm so sorry. We have to stop there. Sorry about that. Thank you very much to the panel. Thank you equally to Luc. Let's now move up to Belgium. We're going to now meet the startup MyNeo and our pitcher, Bert Kussens. So Bert, if you're ready, you have 5 minutes to pitch.
Perfect. Hi, everyone. My name is Bert Kussens. I'm the Chief Operating Officer at MyNeo. Our mission is to make immunotherapy a reality for every cancer patient. Cancer immunotherapy has gained a lot of momentum in the past couple of years because of the success of immune checkpoint inhibition therapy. But the reality is that it's still a small fraction of all cancer patients that can benefit from this wonderful therapy. Even when eligible, a lot of patients do not gain any clinical benefit from immunotherapy. The key to this problem is neoantigens. Neoantigens are tumor-specific immunogenic targets that the immune system of a patient can use to recognize and attack tumor cells and eradicate the tumor from the patient's body. However, finding the right neoantigens to target and load on the immunotherapy is challenging for a number of reasons. One of the reasons is that, as are the patients, tumors are genetically unique, and often there's quite some genetic heterogeneity within the tumor itself. But it's very crucial to load the right neoantigenic targets on an immunotherapy because you don't want to overload a patient's immune system, and you want to obviously avoid any possible autoimmune adverse events. Luckily, there's a lot of omics data available, as well as novel artificial intelligence methods that can help us solve this problem. And that's exactly what we did with the Immuno engine. It's a cutting-edge computational platform for neoantigen selection that, based on tumor whole genome sequencing analysis data, investigates a broad spectrum, a broad landscape of possible neoantigens that can be targeted, but also prioritizes those neoantigens and selects the combination of neoantigens that will trigger a strong immune response and lead to maximum clinical benefit. We've developed and support this computational platform with a world-class team with over 100 years of cumulative experience in genomics oncology and immunology. We come out on top in benchmarking studies when we look at the prediction performance of our platform and comparing with competing systems. But we've spent a lot of time and effort in making sure that our workflow is actually compatible with clinical timelines, and we focus solely on clinical outcome and optimizing the clinical outcome for individual patients. But apart from that, we work with multiple biopharma partners to make sure that, first of all, we gain valuable data out of those partnerships that can help us continually improve our platform, as well as make sure that we remain unbiased with respect to the underlying therapy platform. Our technology plays a key role, is a key part of the final therapy that will be on the market and will set up that therapy for success. That's why, as a business model, we've chosen to license out our technology to biopharma partners in long-term licensing deals. With this model, we've already been able to sign quite some co-development contracts, co-development deals with a number of companies. We have a very strong pipeline going forward. We aim to hit between 400,000 and 500,000 euro in revenue this year, but our strong pipeline will allow us to at least double this in the years to come. We're currently looking for 3.5 million euro in revenue to become the #1 partner to biopharma worldwide. And I'd like to end with this last slide on our opportunity. If we're able, if one of the deals that we plan to sign in the coming years will materialize into an on-market therapy, and we prescribe this to a couple of thousand patients, which is about 1% of the patients currently receiving immunotherapy annually, this would translate already in an annual revenue for myNEO of over 100 million euro. Thank you very much.
Thank you very much, Bert. Thank you for this smashing pitch. Great, great stuff. Let's go to the panel, to the jury, to open our 5 minutes of questions. I see that Alain is raring to go. Off you go.
Thank you for the presentation, very interesting. Perhaps you could tell us a little bit more about the data that's driving the key weighting in terms of the decision-making process around the technology.
Yeah, sure. There's 3 elements to this. First of all, we start with whole genome sequencing data from individual patients to find neoantigens, which makes sure that we have this very broad landscape of neoantigens. And that makes that we can find neoantigens even in tumors that have a very low mutational burden, that have very few mutations. Secondly, there's 2 elements to the prioritization and selection of our neoantigens. One is, and both of them actually mimic what happens biologically inside the cell and with the immune system. One is predicting presentation likelihood, and for that we have amassed quite some both public and privately generated mass spec data to train our algorithms. The second is around immunogenicity, and there too we rely obviously on highly curated and structured data, public data that is available, which we've annotated painstakingly over the past years, as well as data from own experiments that we designed, both for training the algorithms as well as for validating the output of the algorithms.
I see Lena. Please go ahead for your question.
Nice presentation. Could you comment a little bit on the competitors on the market and whether there's similar technologies and what really differentiates you from them?
Yes. Yeah, there's a lot to say about that. I'd say I think the company that comes closest in terms of competing with us from a performance point of view, from a technology point of view, is OncoImmunity, who was acquired by NEC a couple of years ago, where others often fall short with respect to the clinical timelines that you need to meet to be able to do this on a personalized level. So it really is our core element to get this technology as good as possible. That's our main mission, our main focus. We're not a clinical stage company ourselves. No, we are here to maximally support the clinical success of the biopharma partners that are developing the immunotherapies. And that's you can see it in all of the differentiators that I listed in the previous slide as well. It's these clinical timelines and making sure that you can offer this type of technology combined with the therapy in an individualized manner for individual patients is key. I already mentioned the whole genome sequencing data, which is a differentiator. So on a technical level, there's a lot of differentiators, but on the business level, let's say, it's the licensing model and the fact that we focus solely on clinical results that makes us unique.
Cillian, I believe you have a question.
Thank you, Bert. I might have missed it in your presentation, but I was wondering, do you, as part of your service, actually also confirm presentation of these neoantigens on MHC complexes? Do you do peptide elution and proteomics or mass spec on that yourselves? Yeah. Or do you do that up to the company that's taking your service?
You have to make a distinction between co-development on the one hand, and then when we go into clinical trials on the other hand. In co-development, yes, we discuss that. We can offer that through partners that we work with to the company that we co-develop the therapy with. On the other hand, when going into the clinical trials, it's actually the process that has been validated. We do not, for each individual patient, when we come up with a set of targets, we do not go and validate those individual targets again, which would not work in a personalized context.
Yes, go ahead. We have 20 seconds.
A very short question. For the co-development deals, do you also get kind of milestones and royalties when the company enters into clinical, or is just for the first part upfront?
No, that's correct. So we typically discuss in the license deals milestones for development, for regulatory milestones, and then commercial milestones down the road. Correct.
Okay.
Okay, thank you so much. We've just run out of time. That's covered everything. Thank you so much. So moving across now to Germany, where we're going to listen to Dr. Sophia Doll from Omikera Diagnostic GmbH. Hello, Sophia. It's your 5 minutes to pitch. The floor is yours.
Thank you. Know it, cure it. My name is Sophia Doll. I'm co-founder and managing director at Omikera Diagnostics. And I'd like to tell you a short story to explain you what we mean with know it, cure it. I almost lost my dear friend Sarah. She's probably the most successful heart surgeon I know of. Definitely workaholic, but also knows how to have fun in life. She was diagnosed with a rare lung cancer type with limited treatment options. And the doctors told her that one sentence that all of us are afraid to hear one day. You have cancer. You have end-stage cancer. Yearly, millions of individuals die of cancer and the harsh treatments they're going through. What makes it very difficult for doctors is that we're lacking reliable diagnostic tests for precision oncology to decide which treatments works best for every individual. In particular, when it comes to rare diseases, doctors are often confronted to a difficult situation like in Sarah's case. Patients want to fight. They want to get further treatments, but have already gone through every standard treatment of care. So you can imagine that together this results in a huge social and emotional impact, but as well financial losses. So what's the solution? It's proteomics, the analysis of all proteins in the human body. Take this simple example of the caterpillar versus the butterfly. They share 100% the same genes, but what makes them different are the proteins. Proteins are the molecular drivers within your cells, and they drive diseases too. So if we want to understand cancer, we need to understand proteins, and we need to understand all of them. At the moment, and for most of our competitors, proteomics tests look like that. One test identifies one protein. With us, it will look like this. In one test, we identified thousands of proteins. We digitalized the entire human proteome, generating a more complete and comprehensive picture of human health and disease state. What makes us unique is our proteomics technology platform. We have set up an industry-grade platform and shown that we uncover new biomarkers in tissue as well as liquid biopsies, giving information about alternative treatment options. This platform is our intellectual property. We have filed 2 patents enabling us to achieve higher sensitivity, higher throughput with a capacity of about 100,000 samples per year. And with our machine learning-based data analysis, we get higher information content than ever before. With our OmicEra test, we will be part of a patient's journey to offer treatment options and predict disease trajectories to every individual cancer patient. Samples collected by physicians will be transported to OmicEra labs where an easy-to-understand report is generated. This report is discussed in tumor boards together with doctors to decide on follow-up as well as treatments. This will result in an increased number of targeted treatments, off-label use of drugs, as well as prevent therapies with low success probability and strong side effects. We have shown that this works. The tumor board approved our epigenetic inhibitor that we suggested to doctors to treat SARA. Insurances also approved our recommendation and even paid for the treatment. So we have made mass spectrometry from a tool for specialized laboratories in academic research to a tool that can be used in a clinical settings to help patients like Sarah. Now we need to prove the clinical utility in a larger patient study. And we have a clear plan how to get there, but need your help. We're currently looking for Europe and US top healthcare as well as oncology experts and investors. We're preparing for a Series A to roll out our precision oncology arm. We can't stop people like Sarah from getting cancer, but we can find out early to get her the treatment and only she needs to live a life that would make her happier. This is the amazing team behind OmicEra. We are fortunate to have Matthias Mann, who is the most cited German scientist, on board. And together with our partner, we all agree, why wait? Let's find out now together to cure it. Thank you very much.
Thank you, Sophia. Thank you. Well done. And thank you for this very interesting pitch. Let's move over to our jury, who I'm sure have got questions. I go straight to Julien. Your question. It's over to you.
Yes, thank you for the presentation. I was wondering if with the technology, you can also basically follow the activation or inhibition of a given signaling pathway, focusing maybe on phosphoproteomics or other post-translational modification. Yes. And compare that, with responder, no responder, or follow the course of and the efficacy of a treatment.
Yes, thank you very much for that question. Actually, in the case of Sarah, we looked also at phosphoproteomics because she also had a Foundation Medicine report. And the really interesting thing is here that if you see that kinases are mutated, and in that case, we also found that the signaling pathway, all the phosphorylation cascade was upregulated again. It really increases your confidence in basically validating what genomics data say on the active part, which is the phosphoproteomics part here. So that's one, phosphoproteomics plays a very crucial role, especially in the case of oncology. But with our platform, we can also look at other PTMs, ubiquitination, methylation, you name it. But that's definitely possible with our platform, yes.
Iris, please go ahead.
Yeah, you mentioned the intention of moving from the lab, from research to more clinical applications, and you mentioned insurance approved. What does it actually means in this case?
Yes, so in that case, it means that, so because of ethical reasons, so this was within a collaboration, so where you look at end-stage cancer patients, and you always need the support of medical doctors. So we will never be able to convince insurances. So this is not on us, but our part is really like Foundation Medicine to provide more information. And that's why we call it this pitch, know it. The more we know about a particular cancer, let it be on the genomics or proteomics level, and we can transfer that information in an easy to understand report to doctors so that they can then make the decision about how to treat the patient, and the doctors then get into contact with insurances about whether this medication, in that case, it was an off-label use, could have been used, and then the insurance is also paid for that.
Okay, treatment. Like decision support tool, not really insurance companies that will be, I get it. Okay, yes. Thank you.
Yes.
Yes, Emma.
Following with Iris' question, then in terms of the business model, can you comment a little bit on that, how you plan to, let's say, who will be your customer base? Is that major hospitals, or will you engage with pharma companies that are offering treatments for those specific patients?
Very good point. And actually, our main customers are currently pharma companies. But the idea would be to increase the part of where medical doctors are actually going to be your customers. In the US, it's easier if the patients are directly your customers, but in Europe, it's really our largest market is really with the pharma companies and a very still small part of doctors or patients.
Thank you. Alan, I think you would like to ask a question. Please go ahead.
Sure. Thank you for the presentation. I was just curious in terms of your next phase, which you're raising the capital for, is to get, to secure additional clinical validation. Could you tell us a little bit about your clinical trial planning or clinical study program?
Yes.
In terms of gathering that validation data?
Yes, absolutely. So what we've done for now is only on a handful of patients to show that we can provide additional information. The next step would really to be able to show that you have a clinical effect. And that, of course, involves a much more larger study where we want to show that you can increase the survival time by at least 3 more months. And for that, there are different ways. I mean, since we're still a small startup, what could think of first starting with retrospective clinical studies. So this would be our very first aim. And then also to piggyback on existing clinical trials instead of starting really our own first trial, which would be much more costly and for now out of range for us.
Good. Would anybody like to ask a final question? A quick one. Okay. Well, thank you very much, Sophia, and thank you equally to our jury for those very interesting questions. Let's now move to Spain for pitch number 4 in our first sequence this afternoon. So we're going to meet Nathan Lobo from Onina Medicines. Nathan, it's over to you.
Great, thanks so much. A question I ask myself often is, why is a cancer diagnosis still terrifying? And it's because I know 2 things are true. 2/3 of the time, it's most likely that my treatment's not going to work. And even if it does work, most often or most likely, it's going to come back with a vengeance. So why don't cancer drugs work well? Well, it's because not all cancer cells are created equal. Current drugs, and from what I can tell, future ones as well, do a great job of killing the easy to get ones. But there are always a handful of cancer cells that are highly malignant, escape the immune system, and hang around to grow the disease back. Those cells are called cancer stem cells. There are no cancer stem cell specific drugs in the world. That's what we make at Onena Medicines. When we were postdocs at Stanford, my co-founder and I discovered a class of powerful growth factors that cancer stem cells specifically use to keep themselves growing immortally. We named those proteins dual SMAD inhibiting proteins, or DSIPs for short. At Onena, we make antibody drugs that target individual DSIPs, forcing drug-resistant cancer stem cells to mature into benign ones. Our first drug, OMED-001, is a humanized antibody that completely neutralizes the prototypical DSIP protein lefty1. The drug structure and use in cancer treatment is protected by Onena's own PCT filing, as well as a co-exclusive license that we're currently negotiating. One of the indications that we're exploring is glioblastoma, a rare brain cancer with poor treatment options. So glioblastoma is one of the world's most aggressive cancers. You would be lucky if you survived this cancer for even 1 year after diagnosis. Importantly for our therapeutic hypothesis, glioblastoma has been well proven to be a cancer stem cell driven disease, especially in the recurrent setting. On the left, you can see data from an in vivo study with human glioblastoma cells. As you can see, the brain cancer cells can easily grow when treated with temozolomide, a standard of care chemotherapy treatment. However, when the drug-resistant cells, they lose that ability when they're exposed to OMED-001. On the right, you can see two representative histology images from control and treated tumors. You can clearly see that OMED-001 treatment completely eliminates all of the cancer stem cells. To build out our pipeline of anti-DSIP drugs, we have a unique drug discovery platform built upon two highly sensitive genetic sensors. Our platform, which we call the Transferable SMAD Sensor System, accelerates the most tedious and time-consuming steps in anti-DSIP antibody drug discovery. The system saves us precious time and money. We aren't the only ones that are hungry for a cancer stem cell drug. There are a number of examples in clinical trials or on the market of drugs that try to kill cancer stem cells. We stand out from our competitors in two major areas. Our drugs, to our knowledge, are the only ones that are truly cancer stem cell specific. And DSIPs are needed by all cancer stem cells, regardless of tumor type. And that's why we can go after the hardest to treat cancer patients, like glioblastoma patients, triple negative breast cancer patients, metastatic colorectal and prostate cancer patients. Onena operates a business model where we discover and develop antibody assets until preclinical or early stage clinical trials, and then partner out those programs to larger pharma or biotech companies. Typically, each program needs about 10 to 15 million euros of upfront investment and 3 to 5 years before they can achieve healthy returns. This is a well-proven model in biotech to achieve an M&A or an IPO exit at about 5 years away from today. Onena's, with about 1.5 million euros to date, we've accomplished a lot. We've advanced a lead drug candidate, we've started 2 more drug programs. We've also deployed our proprietary drug platform. We're looking to complete a seed raise to drive our first program into early preclinical studies, as well as advance our 2 other programs in a staggered fashion. Onena's management team has a lot of logos from a lot of great institutions. We have made drugs before and we have gotten them into clinical trials. Our advisors have even more logos associated with their experience. I just want you to remember one thing. We make cancer stem cell specific drugs.
Nathan, I am so sorry, but time's up. Can we, thank you so much. Thank you very much indeed, but I have to play the game. Let's now listen to the questions. Yeah, let's now listen to the questions from our jury. I open to whoever would like to go first. Renato, please go ahead.
Yes, thank you very much for the presentation. What is the use of proceeds that the capital you are trying to raise?
Yeah, we're trying to raise a total of 2 million euros of seed, and that's going to accomplish 3 tasks. The first is to get our first program into non-GLP tox studies, our second program into lead candidate status, and our third program to identify a candidate out of a hit to lead campaign, and then fully mature our platform and deploy it across all of our drug discovery efforts.
Very interesting target, and I mean there are some other companies looking at this target as well. I guess just to follow on from that question, it's a very small amount of seed capital. You've got a lead and then potentially 2 pipeline programs in a highly competitive, quickly evolving space. So certainly my suggestion would be that you look at that again, and if you've got a real opportunity, a really interesting target, to ramp up that raise and to do more. I think you're not raising enough capital, perhaps not being ambitious enough in the context of a very interesting target.
Yeah, I appreciate those comments. Our goal here, we recently have been selected as the next space launch company, and the LPs in that fund are Roche, J&J, CSL Behring, Purius BioVentures, Roivant, and BridgeBio out of Korea. And so we already know that we can partner with those companies to achieve a really healthy Series A. And so really this seed capital is really to get that lead drug into early preclinical, get the proof of concept done that we can manufacture it, and then raise a larger Series A. So this is really, to cover the time frame for the next year. And, we're supplementing that with grants as well. So I take your point that a lot of capital is needed for therapeutics. And we'll keep that in mind.
Yes, Cillian, go ahead.
I was just wondering about the antibody approach. So I guess for approved antibodies like aducanumab, brain penetrance is only about 1 or 2%, right? So it's hard getting these antibodies into the brain. And you run into all sorts of risks when you have to increase the dose high. With other small molecule approaches also in the market, could you comment on why you went for an antibody approach and why you think it will be better?
Absolutely. Thank you so much. So the 2 questions about getting it into the brain. The first is there are clinical trials that have just been done with nivolumab that unfortunately failed in glioblastoma, where patients can receive drugs directly injected into their tumors in the brain. So you can get around the blood-brain barrier that way. And the second is there are companies like Denali out of the US and San Francisco that have technologies to cross the blood-brain barrier and chaperone antibody drugs across. Okay, but that would require some deal making. In terms of small molecule approaches, so the activity that drives the cancer stemness is the interaction of the protein with a number of cell surface receptors. In order to stop that binding, small molecules aren't big enough to interrupt that interaction efficiently. So this type of approach is well proven to neutralize the activity of secreted molecules. And I'm actually not thinking of aducanumab. I'm thinking of something like Humira. Well proven strategy in the clinic is what we're really trying to model our drugs after.
Thank you. Julian, let me bring you in here.
Yes, thank you. So I guess the good thing with glioblastoma is we have so much nothing that even a little improvement would make a big difference for the patient. But coming back to that type of tumor, it's quite heterogeneous. So have you looked at different tumor samples or working with primary cells to test your antibody?
Yes, both. Both. This approach works on both patient-derived xenograft patient samples directly taken out of the clinic and never touched a mouse. It works on cell lines that are derived from glioblastoma that you can buy from, commercial databases or academic databases. It seems to be a broad approach that's applicable to the way cancers grow in general, not specific to the models that we're using.
Okay, thank you.
Sure. And it works on breast cancer.
We're just running out of time. Thank you so much, jury, for those very interesting probing questions, and to Nathan in Spain. So this brings us to the end of our pitch session number 3. And let's stop a second just to remember who we have met so far this afternoon. We first met Luc from Limula in Switzerland, who are saving lives by enabling widespread access to the latest generation of personalized cancer treatments, bringing automated manufacturing of cell therapies to the point of care. Then we went to Belgium to meet Bert from MyNeo, the world leader in genomic-informed drug discovery, on a mission to help unlock the potential of immunotherapy for every cancer patient. Then we went to meet Sophia in Germany at Omicera Diagnostics. By reinventing blood tests, Omicera is dedicated to improving health by proteomics, catalyzing the discovery of reliable and novel disease biomarkers. And finally, Nathan in Spain, who are making antibody medicines that force drug-resistant cancer stem cells to permanently grow old, to no longer sustain and spread cancer. This is Onina Medicines. Thank you to all our pitchers this afternoon so far. Well, we're just under 3 hours now before the ceremony awards, and we have one pitch sequence to go. If you are interested in finding out any more about any of the startups we have seen this morning and so far this afternoon, then our network platform is definitely the place to go. But before that, and before our next session, we're going to take a short 20-minute break, starting back at 14:30 Central European Time, 2:30 sharp, to have our fourth and final pitch session. So we look forward to seeing you then. Have a great networking time.